Hi, this is Mr. Worski, and today we are going to start applying the information that we have, started filling in some holes so that we can get a clearer view of our genome and how our genes are regulated. But we're first going to start with molecular genetics of Proks and viruses. Now, if you have any questions, watch the video over again, check out your textbook, or even ask me in class. So what we're going to take a look at first right here is the DNA, and the way that the proc and the euc setup is going to be the same. All right, so we're going to talk about DNA for both, but we're going to focus on procs right now. So the bacterium, we know that their DNA is in the form of a singular continuous loop. It has about 4.6 million DNA bases, which is about 4,300 genes. It's a very small genome. Now, it's about 1,000th, 1 in 1,000th the size of our cells. Many prokaryotes have plasmids that are about 3 to 12 genes in length, and we have a picture below here of a nucleoid as well as some plasmids. So that's the DNA. We're going to talk about DNA. We're going to talk about DNA replication. Now, DNA replication looks like the Greek letter theta, and the Greek letter theta is a circle with a thing in the middle like that. So it's called theta replication. The OREC is the origin of replication, which is here and here. There's a place. So helicase unwinds the DNA at that spot, and then DNA synthesis occurs bidirectionally. It occurs from a 5' to 3', and a whole bunch of 5' to 3', the lagging here. So we have this DNA synthesis forming here, this DNA synthesis happening here, and they will eventually peel off and form two loops of DNA. But the DNA replication in prokaryotes is called theta replication because it looks like the Greek letter theta. It starts at the origin of replication, which is OREC, and it happens bidirectionally with both leading and lagging strand synthesis. Now, remember, this is adding to your knowledge. You should know some of the material being discussed here. So, transcription. Okay, that's how the first step in how we produce proteins. So the structural gene, the segment of DNA that codes for protein. Hopefully we got that. We understand that. A section of the DNA that codes for protein or a polypeptide is called a structural gene. Now in prokaryotes, remember that in protein synthesis, hnRNA is not produced. Instead, mRNA is produced right away. So there are no introns in prokaryotes. RNA polymerase binds to the promoter, which opens about 10 bases at a time, produces mRNA at a rate of about 45 nucleotides per second, which is really quick. Here's a picture of prokaryotic transcription. We have initiation, which the RNA polymerase lands on the promoter. It opens it up about 10 bases at a time and produces the mRNA. Beautiful. Now, as this happens, a ribosome can attach to the mRNA and start producing proteins in translation. So the prokaryote is really efficient in that it does not produce all proteins all the time, right? Not all proteins all the time. So it regulates the production of proteins. And how it does that is that it uses something known as an operon. So that is how a prokaryote can control the production of proteins, is through an operon. Eukaryotes have a larger number of ways to control protein production, and we're going to talk about those later. In an operon, we have a section of DNA. We have the R, which is called the regulator gene. It produces a protein called the R protein, which will repress or stop protein synthesis. And that's somebody that now the dotted line here, it represents a number of DNA bases that you don't know how many, it can be far away, it really doesn't matter. We have the P, which stands for promoter, and we know that the promoter is where RNA polymerase lands. The operator, right here, this is what the operon is named after, the operator. That's your on-off switch, and then you have structural genes. And you can have any number of structural genes. You have X, Y, Z, whatever. Okay, draw that picture down there. We'll be referring to it from time to time. So here again is the operon picture. We have the regulatory gene over here somewhere. This is a section of DNA, any distance, promoter, operator, structural genes right here. Now the structural genes, when they're read by the RNA polymerase, will produce proteins A, B, C, and D, and so on. So the operon, the repressor, which is the R protein, will bind to the operator, and it overlaps into the promoter region, and that will stop RNA polymerase from landing. So if there is a protein on the operator, protein synthesis will not happen. No protein is produced. So if the repressor is on the operator, the operon is off. So here we have the repressor protein. It's on the operator. Notice it overlaps into the promoter. RNA polymerase cannot land. It cannot transcribe the structural gene. So now there are two types of operons. We have inducible and repressible. We're going to talk about the inducible first. The repressible is the opposite of the inducible. If you understand the inducible, you're all set. Now they focus their attention usually, College Board, on the inducible operons. In the inducible operon, the operon is off. So that means that there is a R protein or repressor on the operator. If there's a protein or repressor protein that is on the operator, the RNA polymerase cannot land on the promoter because the protein is overlapping into the promoter. If RNA polymerase cannot land, you cannot produce a protein. You cannot produce mRNA, you can't have translation, no protein is produced. So an example of this is a lac operon. But first we have to talk about an inducer. Now an inducer will bind to the repressor protein, and when the inducer binds to the repressor protein, the shape changes and the repressor can no longer bind to the operator. When that happens, then it floats away and the RNA polymerase can now land on the promoter and you can now produce a protein. The lac operon is an example of an inducible operon in that we have allolactose, which is an isomer of lactose, is the inducer and will bind to the R protein. The inducer or allolactose will control the production of lactase or beta-galactosidase, which is another name for lactase. Now, this lactase will always break down lactose into glucose and galactose. And here's a diagram. Okay, so we have the repressor protein here. Okay, it's bonded to the operator. This right here is allolactose. It should be. It binds to the inducer, which changes the shape of the R protein. That unbinds. RNA polymerase can land on the promoter, which is the blue here. This right, or if you're colorblind, it's right here. This is the operator. RNA polymerase will now produce mRNA for the gene lactase. Here's another lac operon thing. Here's RNA polymerase. It's landed on the promoter, but it can't get anywhere because of this R protein here. The inducer binds to the R protein, changes the shape, leaves. Now RNA polymerase can land and produce the mRNA for these genes here. So that is the lac operon. Now the repressible operon is the opposite. So if the inducible is always off until it's turned on, the repressible is always on. So they're always producing proteins until it is shut off. So that means that a repressor protein needs to bind to the operator. So the repressor is floating about. Something binds to the repressor, which changes its shape, and then it can bind to the operator. If it binds to the operator, RNA polymerase cannot land on the promoter. It cannot produce the mRNA, and no protein is produced. An example is tryptophan synthesis. Tryptophan is an amino acid. Right? There's a multi-step reaction. When tryptophan gets too high, the tryptophan actually binds to a repressor, and that binds to the operator, and that stops the formation of tryptophan. So this is known as an anabolic process in which you're building stuff up. Remember, too much of anything is bad. So this is one way they can regulate the amount of tryptophan in the cell. Now, when tryptophan concentrations get low, tryptophan falls off, the protein changes shape, the repressor changes shape, leaves the operator, and then the production of tryptophan continues. Production of the tryptophan enzyme, or the enzymes that make tryptophan continues. So here's the trip operon. It's producing these things here. The tryptophan binds to the repressor, binds to the operator, cannot produce, RNA polymerase cannot land and produce the enzymes necessary. Pictures would really be good. Get them down. So now we have the CRP, CAMP, and I call it crap camp. Of course, I always picture a crap camp flags. Welcome to crap camp. But CRP is CAMP receptor proteins, and CAMP is cyclic AMP. Now, cyclic AMP is produced when you break down ATP to ADP, that's adenosine triphosphate, to adenosine diphosphate. When you remove the phosphate, 7K calories of energy removed or released. If you break down ADP further into AMP, then 7K calories of energy is also released. But the problem is that all the energy is used up when you have CAMP. So when the production of energy is low, CAMP is produced, right? Because you're breaking down ATP into CAMP. That binds to CRP, and that complex, the CRP CAMP complex or crap camp complex, will bind to the promoter and it acts as a flag to attract RNA polymerase. Now, this will increase transcription by 20 to 50 times. So we can tie that into the lac operon when lactose, the energy. So if the bacteria only is exposed to lactose, it needs to break down glucose into energy. Problem is lactose isn't glucose, so we need lactase to break down lactose into glucose and galactose. How do you do that? The allolactose will bind to the repressor protein. It removes the repressor protein. RNA polymerase can land, produce lactase, which can now break lactose into glucose and galactose. Meanwhile, the cell is using up all its ATP. If it uses ATP, CAMP is being produced. It binds to CRP, which lands in the promoter, attracts more RNA polymerases so you can produce more lactase, so you can produce more glucose, so they can produce more energy. And here's the crap camp diagram. See right here, it attracts RNA polymerase and it produces the genes necessary to produce energy. So now we have movable DNA. So DNA, you might be thinking is plastic, but it is not. It can move. Transposons are known as transposable genetic elements. It's another name. And there are pieces of DNA that can move from one place to another. They're also known as jumping genes that were discovered by Barbara McClintock, where she earned herself a Nobel Prize. The ends of transposons are either repeats or inverted repeats, and they're anywhere from 20 to 40 nucleotides long. There are two types of transposons. We have simple transposons with the ends, and then you have a gene for transposase in the middle, and that gene is what cuts the DNA and allows it to either jump or insert into genes. Now, if it inserts into a gene, it's going to inactivate the gene and cause a mutation because it won't be the right shape anymore. So transposons are not good at all. They will only turn off genes if they land in the middle of them. We have complex. Now, complex transposons are two simple transposons, and then in the middle you have genes. So the complex will only work if the two simple transposons jump together, and then it can carry all the stuff in between with it. Now we're going to stop. Oh, we are done. Look at that. We are done with molecular genetics for prokaryotes. Now, if you have any questions, please review the video, read your book, or ask me questions tomorrow. Have a good night.